primary objective is really to look at pharmacokinetics and safety and tolerability of single and multiple doses of the drug. And the secondary objective is usually pharmacodynamic. So the pharmacodynamic markers should confirm that the target is hit, that, and if possible, help select the dose for the next level of study. Good PD results are very helpful because they encourage investigators to enroll patients when you start going into the phase 2 patient studies. Now, there are two basic designs that can be used in these EIH studies, forced dose ascensions versus multiple dose crossovers. The sequential forced dose ascension is basically you have a different group of people for each dose level. So if you have 5 dose levels you're testing, then you have a different group for each one, and you test the lowest and finish that before you test, so test group 1 first, then group 2. When you finish group 2, you go to 3, and so on. So it's all so you don't have any big safety surprises. Later trials are done with what's called a parallel design, which means all the dose groups will start at the same time together. That's done in phase 2 and phase 3. Crossover studies are when you have the same group who comes back for multiple dosings. And the big difference in these two are that the sequential forced dose ascension it'll look at more people, so you get a fresh group of people. There's no bias being carried over from a previous experience of the drug, and whereas the crossover tends to be very sensitive, because you're using each person will get placebo and at least 1 active or 2 active doses. So if there's a real tendency for that drug to do something, it's much easier to see. And it's very, very useful in cases where you have a potential arrhythmia liability for the drug. It's also a little bit cheaper because you don't have as much recruiting cost, and but there are two big problems. One is the dropouts, so you have to have a little bit larger group size. And then the other thing is what are called carryover effects. So if a drug has a long half-life or it has a long sort of pharmacodynamic effect, these create a lot of complications for that design.
What's a typical waiting time?
5 half-lives of the drug. So, and it's a little tricky to do it in the SAD because you don't know what the half-life is yet. And it's not just the half-life of the parent, but it's also the half-life of the metabolite. So usually you study healthy volunteers, and but you can study patients. But and a lot of very small companies, or not very small, but small companies, can really try to study patients, but it's not without its problems. You have to recruit patients. That means that it takes time, and you also have to go to multiple centers, which means the variability around the trial starts going up. Patients are on concomitant medications, and particularly patients who have active disease, which is the ones you'd really like to study if you're going to see efficacy. So you now have these concomitant medications potentially fouling up the PK or the safety around the new drug that you're testing, and you can skew the safety profile. So if a population of patients, say, typically has a lot of liver problems, in your first study, you're going to have liver problems. And if you went into healthy volunteers and had no liver problems, you have a cleaner compound. You may still have liver problems in that patient population, but at least you establish a clean baseline. The other thing is that the number that you need for all these pharmacokinetic measurements is actually not too large. It's only about 6 people or so. And when you get into patients, you tend to want to see numbers up more in the 15 to 40 range. And so you get a very incomplete efficacy determination. You can see some useful PD rule readouts because disease pathways are activated in patients. Patients are clearly more relevant than healthy volunteers, and patients may tolerate more risk. Again, if you go cancer patients are the classic thing where often they will go in there. Number of subjects, often a group size will be 8, with 2 of those placebo and 6 active, or you can put 8 active, but some number in there. Usually these groups are somewhere around 8. And for a 5-dose level study, which is fairly typical, you'll go to 50 or 60 patients. The blinding in this is a little different than the later trials. So blinding is just who is aware of what the patient is receiving. Here, the patient and the observer, the person scoring any test that you're doing on the patient, are usually kept blind. But the drug is actually made at the site by the pharmacist. And that person will know actually what the patient is getting. On the company level or the monitoring level, the sponsor, usually you may all be blinded, or you may allow certain people to be unblinded. And the drug supply people know, obviously, and then the statistician and the clinical pharmacologist. And that enables the company to look at data as it comes out of this trial and to adjust the trial in a way that would, make it more precise, if you will. The drug product usually for small molecules is suspension because it's the most flexible form, and you can change it pretty easily just by putting in more or less powder into the vehicle. Now, there's some more on design here. This is the dose. This is really dose selection. So as I said before, it's usually 1/10 of the NOEL as the first dose. Top level is more variable. I mean, you really don't want to go above or up into an unacceptable toxicity. To range up to where animals have been dying, particularly if they're acute deaths, is not very well. If they're acute deaths, it's another whole problem. But you really don't want to go there, and you do not want to exceed the top of the tox exposures. The highest amount, you can set it at the highest amount that could be given for an indication, but that's not a very good thing to do. You really need to go above that because you need to for some subsequent required studies. So the basic thing is you push your dose as high as you can, and you stop either when you have unacceptable side effects, adverse events, which are called AEs, and or you reach your highest planned level. So you typically, for a small molecule, you might try to go up to 2 to 3 grams for a sort of classic organic. You wouldn't go that high, say, for a retinoid or a steroid. I mean, those will tend to be lower, but garden variety stuff, get it up. The step size range is usually around 3. You go up in steps of 3, but you can go anywhere from 2 to 10, depending sort of how big of a dose range you have to test. The centers, which is where the sites are conducted, they're generally in single centers unless patients are involved. And then if patients are involved, it depends on the recruitment rate and how many patients you need and how long you want to take to do the trial. Assessments are, of course, what you measure in a trial. And the safety assessments are pretty standard. They're hematology, urine analysis, serum chemistry, ECGs, all that stuff, vital signs, physical examinations, and so on. Extra safety assessments are based on toxicity. So, for instance, if someone, if let's say the animals are kind of keeling over at T max, you might ask people to walk heel to toe in a straight line for 5 meters or something, sort of like a DUI examination. And this is important, obviously, because if there's a problem, when you get to studies where the patients are taking the drug at home, you need to precaution them about operating machinery or driving or so on. And the times are, sampling times are fairly frequent for PK, but they can be adjusted if you find that the PK is a bit different than what was thought. The most common thing here is the drug hangs around a lot more than thought, so you have to add additional time points at the end.
So are the safety assessment tests, they're becoming more sophisticated now with biomarkers and so on? I think that's kind of the direction of the
Well, that's a long, that's a big area in and of itself. And the problem with safety is you have to interpret the data. And for a unique biomarker built around a particular compound, it becomes hard to do. There was just in June of 2010 an issue of Nature Biotechnology that is devoted to biomarkers for renal disease. And they are advocating a whole set of markers that they claim to have validated in animals, and they suggest that they should be used in human screening. And I actually think that recommendation is premature. It probably should be done, but I think there's going to be big questions about interpretability. Even things used in animals, if you look at things like the Hurg assay, when that came in, it took about 5 years to figure out how really to interpret it. And I think the same thing. So in general, you should target biomarkers much more for efficacy and leave safety in the hands of well-validated assays, because you can't interpret. And where it's not just, I mean, you've got to know whether other drugs interfere with these tests, and it's too complicated. Anyway, let's not get off on that. The PK, what happens at the end of this? How do you evaluate it? The observed exposures, you should have covered the projected efficacy range, and you should have exceeded it at the end of it. But it should have been done with an exception